So I guess the first thing I wanted to is motivate why would you need to build a translational dream team? And I think the big thing here is translational science, moving things from the bench side, from the bench to the bedside, has become a big focus of the NIH lately. And so I wanted to walk through an example that tries to motivate this question. So an example that's hopefully germane to most of you, and that is trying to get more funds. So this is an example of an NIH RFA from the National Institute of Mental Health, where there's the Silvio O. Conte Center that specifically says we're interested in applications that talk to us about how you can do translational research in an interdisciplinary and integrative way. And the reason I chose this one, it was from the National Institute of Mental Health, was that my background from a research standpoint is in the area of neurodegenerative diseases, but unfortunately the main ones are Alzheimer's disease and Parkinson's disease, so maybe not that interesting to the pediatric-focused folks here. And but a few months ago, we started working with the one of the largest private funders of autism research. So what I did is I said, let's get an example of an RFA where an institution with some expertise in the area of autism would be interested in applying for, that requires the building of a translational and interdisciplinary team. So I found this example, and then just to motivate it a little bit further, this particular grant pays out at a maximum level about $2 million a year. So while it's not a mammoth grant the way the CTSAs are, Clinical Translational Science Awards, it's nothing to scoff at. And so there are obviously a lot of people who are trying to get more funds to create more informatics positions, hopefully. So after I'd found this opportunity out there and I knew I was going to use it for my talk, I wanted to see who out there who has been successful in winning this particular award. And it turns out right down the street, you have the Silvio O. Conte Center at UPenn. So it also had a local significance to it. So now I just take you briefly through our approach to text mining. It's not nearly as sophisticated as Watson, but it has some things in common. So the first thing you need is some source text, and typically we use papers or grants, but if you are more clinically focused, you might think about abstracts from clinicaltrials.gov, from practice guidelines, or whatever type of text that you have that's written in a somewhat kind of formal and scientific manner. The next thing is we take a thesaurus or taxonomy or controlled vocabulary, and using natural language processing, we index the text using that taxonomy. So we use the medical subject headings typically for when we're working in the biomedical scientific area. But depending on what your application was, I could imagine people being interested in indexing text automatically with ICD codes, CPT codes, or SNOMED. And the output of this is what we call a semantic fingerprint. So on the right-hand side, you can see a list of concepts, and underneath the concepts is a blue bar. And the length of the blue bar represents the importance of that term to the text. And there are a few things going on that determine the length of the bar, the frequency of the term within the text, but then also the uniqueness of that term within the entire body of literature that you're indexing. So once you've indexed a bunch of documents, like if you've indexed all of PubMed, you've indexed all of the NIH reporter database or whatever it is that you're looking at, and if you can connect those documents to people or to institutions, you can start to aggregate these semantic fingerprints. And so, for instance, this is the aggregated scientist-level fingerprint. So this is a well-known autism researcher, and looking at all his funded grants over his entire career and all the papers he's written over his entire career, this kind of represents what his research focus has been and what his expertise is. You can see that the number one disorder is Angelman syndrome, while autism is further down. And the blue bar is based on publications, with the red bar being grant support. So in this case, he's publishing a huge amount of papers about Angelman syndrome, but he's not getting a huge amount of funding from Angelman syndrome, whereas autistic disorder, he's publishing a moderate amount, but he's really getting a lot of funding from it. And seeing things like this it doesn't try to explain causation, but it could hopefully stimulate someone looking at this to ask some questions like, well, maybe could it be because Angelman is a couple of order of magnitude a rarer disease than autistic disorder? Could it be that that's his passion, but he can't get enough funding to support his lab, so he subsidizes it through autism research? I don't know, but they're questions one could ask. The other important piece of information that we mine from the literature are co-author connections. So I took, about 10 or so of the presenters at this conference who have very big co-author networks or relatively big co-author networks, and I created a visualization of how they're connected. So, for instance, Lehman and Abbott are at Johns Hopkins, and they have a few authors in common, or Fix and Grunmeyer from CHOP have a few authors in common. But the one I wanted to bring up, just to kind of give you a little use case of how this information can be helpful, Sittig and Canry have never written a paper together. There's no line connecting them directly. But they have two co-authors in common, Cooperman and Miller. So let's say that Dr. Sittig had to fly in, give his talk, and then get back to the airport and couldn't stay for any other presentations. But someone he knows attended Dr. Canry's presentation and told him, the stuff that Dr. Canry's doing is really complementary to the stuff that you're doing. It's too bad you missed the talk. I think you guys should collaborate. Well, Sittig, maybe, I always like to say scientists are people too. You don't, there's some social inhibition in just cold calling people and asking them if they want to work with you. So if this information were readily available, could call up Cooperman and say, well, you've worked with Canry before. What do you think? Would we be a good pair? Is our work really complementary? And if the answer was affirmative, would you please make the introduction? So now I'm just going to briefly talk about how funding agencies have used this technology to help them categorize their research portfolios, so the people that they fund and the research that those people do. So this is from the NIH. They have a public website where they report on 250 approximately different disease categories in terms of their extramural funding. So this is the one for autism. And the way they do it is the fingerprints that I showed you, they have a category fingerprint for each of the 250 diseases. So these are the terms that are contained within their definition of autism. And every single funded NIH grant gets fingerprinted and compared to the 250 category fingerprints. And if there's a close enough fit, it gets automatically categorized as being of that disease. And any grant can be categorized as being one or more diseases. So you could have a grant that's aging and Alzheimer's disease and anything else where it's a fit for their categories. The other thing I want to bring up is we have a number of customers in the UK, funding agencies, and they wanted to categorize not by disease but by position on the bench to bedside spectrum. So on the left-hand side, you have underpinning mechanisms, etiology, and then moving further right, disease management, health services research. So you can see kind of the various funding agencies, where do they fund on the bench to bedside spectrum. And so they asked us, they said, boy, this is really manual. We have all these experts that spend all this time coding these grants. Would you be able, since you have fingerprints of our grants, would you be able to use machine learning to help us? And we said, actually, we think we would. And so the technique that we settled on was this thing called support vector machines. Historically, a lot of machine learning looked at the typical example of something, and it didn't do very well on the border cases where you're not sure, even a human wouldn't be sure, should I put it in this category or this category? And the nice thing about support vector machines is they focus almost exclusively on those boundary cases, and they try to find the best line that separates the tough-to-call cases And once they've done that, they have like a conceptual map of all the categories, and then they can just figure out where any new document that you need to categorize falls within that map, and then predict the best category for it. So now I'm going to take us back to the original question of, we want to take advantage of this funding opportunity. So in some cases, you'd have someone who's pushing a big funding opportunity regarding a lot of investigators. They might have a clear conceptual map of what that would look like. So on the left-hand side, going down, I have different lines of investigation. On the right-hand side, kind of showing a potential way to look at translational science in terms of the organisms that you're working at. So fruit flies and zebrafish in the middle on the one side, knockout mice and songbirds in the middle, and then rhesus monkeys, and then finally humans in clinical trials. So if you had a very good mental map of what you were looking for, then you might be able to fill in a lot of the names, and you might just have some blanks that you needed help filling in. And so using fingerprints, like I showed you of the autism researcher, we could help fill in. You know, if you're looking for an environmental neurotoxin specialist who's working for zebrafish, we could give you 3 potential gap fillers using these techniques. But what about when you don't know enough about the field? You just know, I got a couple really bright scientists at the bench and a couple who are seeing patients. How do I create this compelling story so I can compete and get this grant funded? So in this case, if we look back to kind of our 2 main elements, you have the fingerprints of publications of people in your institution, and then you know who those people collaborate with. So if we put these together, we can connect people. So this is Larry Zapersky. He's a fruit fly researcher at UCLA, and Eric Hollander, who sees autistic patients at Einstein in the Bronx. And these are all the way they're connected through co-author relationships. So kind of like the Kevin Bacon thing of scientific publishing, how many degrees of separation from Kevin Bacon. So if we take a look at the papers that connect these 2 people, then you can start to get a sense for what are these possible stepping stone categories that could connect them. So then you can cluster all these papers which connect them in these co-author networks into semantically similar categories. Once you've done that, you have the ability to look at NIH funding within these new clusters that you've created. So this is the funding profile for the autism and neural stem cells cluster. And you can see that it doubled from 2001 to about 2007, and then it started to tail off a little bit. So you can say, is this, if I put this in my grant as a stepping stone that connects my basic scientist and my clinical scientists, is this an area where people are successfully getting NIH funds? Because you have to kind of, you not only have to have a bright idea, it has to play in Peoria with a given study section. So then, So finally, you've kind of looked at the various stepping stones, and now you've been able to fill in that Excel spreadsheet in terms of what you want your columns to be, what you want your rows to be, and the only question is which scientists do you want to slot in there. So once again, you can rank people based on their publication history or their success in getting NIH grants in a given translational stepping stone area. But then again, what's important is never to lose track of who has co-authored papers with whom. So who's already proven that they can work together, that they like each other, because just throwing a bunch of scientists together in a big team isn't necessarily an ingredient for success unless there's some social glue or professional glue which will allow them to write a compelling grant and then do good research to hopefully 5 years after the fact have that grant renewed ad infinitum. That's it. Thank you.